The overall goal of this procedure is to analyze in silico restriction enzyme present in a recombinant vector to which it was cloned and inserted to characterize the vector by restriction enzyme and the interpretation of the results of restriction in an electrophoresis yield.
Restriction enzymes are also known as endonucleases and are capable of cutting phosphodiester bonds of genetic material from a recognized sequence. There are three types of restriction enzymes. The first type has a restriction and modification activity. They cut at some distance from the recognition sequence and cut far away from the recognition sequence, upstream or downstream. They need ATP for moving along DNA molecules since the recognition site till the cutting sequence. Its application is to generate fragments for being used as label proofs in southern and northern blotting. The second type, they only have restriction activity. They cut inside the recognition sequence. They only require magnesium. They do not require ATP. Its application, fragmenting genomic DNA for separating it in an electrophoresis and southern blot. Disease diagnosis for allele screening. The third type, restriction and modification activity. They cut 5 to 8 bases before and after the recognition sequence. They need ATP for moving along DNA molecules since the recognition site till the cutting sequence. Its application is to generate fragments to be subcloned into appropriate vectors in order to have recombinant DNA.
In order to create a restriction enzyme map, you must have the sequence of the plasmid and that one of the insert. This procedure should be done 3 times, one with the plasmid sequence without the insert, another one with the insert, and a third one with the insert in opposite clockwise direction. After having the sequence, enter the NavCutter website, paste it, and select the circular sequence option and click Submit. On screen, you will see all the restriction enzyme sites that can be recognized in the sequence, and if you click in Custom Digest, you could select the restriction enzymes of your interest. Those would be the ones that you have available in your lab, for example. Click in the green button that says Digest, and the map of the sites would be shown. If you wanted to see the specific sequences and the place in which the enzymes cut, click on Enzymes and Sites option located in the right lower part of the screen, and a list would be shown, just like the one shown on screen.
For a successful enzymatic restriction reaction, it should be considered the quantity and quality of DNA, quantity of the restriction enzyme to use a lower concentration of enzymes so that the glycerol concentration can be avoided. A glycerol concentration higher than 5% induces star activity. It's needed to select a type of buffer that have a high ionic force between 100 to 150 millimolar. It's needed to control the temperature and time of the reaction. DNA must not contain salts and organic solvents like ethanol or phenol due to its relation with enzymatic activity inhibition. Magnesium must be used as divalent cation.
Once plasmids belonging to white and blue colonies were extracted by alkaline lysis, the following protocol was considered to degrade the plasmid with certain restriction enzymes, ECO R5 and SCA1, in order to detect the presence of the fragment of interest, in this case the ZFY gene. Place all reactants needed in a 0.6 milliliters microtube for performing a 20 microliters total volume enzymatic restriction reaction with the adequate restriction enzymes. After 6 enzymatic restriction reactions were performed, microtubes must be incubated for 1 hour at an adequate temperature, depending on the optimum temperature of an enzyme.
An electrophoresis yield was prepared and run with the conditions shown on screen. In the following table, it can be seen which sample was loaded in which well. Once the restriction enzyme analysis was done, the enzymes that were selected are SCA1 and ECO R5. SCA1 cuts inside the insert, and this gives us information about the direction. On the table shown on screen, it can be seen the characteristics of both enzymes and of the buffer that was selected.
In silico, electrophoresis were done regarding restriction enzyme reactions. We have the gel electrophoresis for the plasmid without insert. Then we have the gel electrophoresis for the plasmid plus the insert in clockwise direction. The plasmid plus the insert with a counterclockwise direction. For the first well, we have a blue colony uncut plasmid. For the second well, we have a blue colony cut plasmid with SCA1. For the third well, we have a colony cut plasmid with echo R5. For the fourth well, we have a blue colony plasmid cut with SCA1 plus echo R5. For the fifth well, we have a white colony uncut plasmid. For the sixth well, we have a white colony cut plasmid with SCA1. For the seventh well, we have a white plasmid cut with echo R5. And for the eighth well, we have a white colony cut plasmid with SCA1 plus echo R5, and last but not least, we have DNA ladder 1 kilobase oxygen as a DNA ladder. The table represents the expected and observed results of enzyme restriction analysis. There are sections in bold can tell the direction of the insert.
Restriction enzymes, also known as restriction endonucleases, are enzymes that recognize a specific sequence of the DNA and cut phosphodiester bonds, like the case of ECHO R1 shown on screen, that recognize a specific sequence of the DNA and cut them. The parameters that we have to take into account in order to make a restriction is to verify the quantity and the quality of the DNA. It's important to use the lower concentration of the enzyme so that the glycerol concentration can be avoided. A glycerol concentration higher than 5% includes star activity. It's important to use a buffer that have a high ionic force between 100 to 150 millimolar. The temperature and the time of the reaction depends on each restriction enzyme. We have to consult the characteristics of each restriction enzyme in order to know the optimal temperature and time of reaction. When applying a double restriction, temperature and buffer must be the optimal ones for both restriction enzymes. When there are two different restriction enzymes, two different temperatures will be needed. In this case, the first enzyme is added and it is incubated at its optimal temperature. Then this first enzyme is inactivated when temperature increases at the optimal temperature from the second enzyme, and then it's incubated. On the other hand, when there are different enzymes in the same buffer in which an enzyme is more active than the other one, the number of units and incubation time can be adjusted for compensing excision ratio.
There are certain conditions, for example pH more than 5% of glycerol, enzyme quantity, organic solvents, ionic force, temperature and time of incubation, in which enzymes may cut in different sites regarding its target sequences. Thus, this problematic triggers unspecificity and obtention of unwanted fragments. In other words, a restriction enzyme could have a star activity. DNA must not contain salts, ethanol, or phenol, since these compounds inhibit enzyme activity.
Blue samples contain topoplasmid without the gene, but if it is light blue, it may contain the gene in an incorrect lecture frame. When talking about white colonies, we have transformed ones, and they do contain the insert in the correct lecture frame, but it can be placed in the wrong direction. The restriction enzyme results could not let us confirm neither the presence nor the direction of the insert. All of the samples that can tell us if the gene was inserted due to the addition of the sizes of the bands that are observed in the gel, but only the following ones can tell us if the insert was placed in the correct direction. White colony cut plasmid with SCA1, but it has many unspecific bands. White colony cut plasmid with SCA1 and DQ R5, but no DNA bands were observed. Without taking into account the lanes in which nothing was observed, in all wells it can be seen unspecific bands that cannot let us analyze and determine whether the insert was or not in the correct direction. This can be due to star activity of the enzymes, and the causes are multiple as they have been mentioned along this video, like high glycerol concentration, high enzyme concentration per microgram of DNA, non-optimal buffer, prolonged reaction time, organic solvents presence, excess of the divalent cation. From those options, the ones that could have been the reasons are the presence of organic solvents and maybe the time of reaction. In the wells that nothing was observed, it is possible that the samples were not loaded properly or that there was no plasmidic DNA in the microtube reaction, and then the enzyme had nothing to cut.
It's important to consider some parameters for performing restriction enzyme reactions such as quantity and quality of DNA. Contaminants may affect or inhibit restriction enzyme activity. Another point to consider in enzyme quantity joined with glycerol concentration, in which enzyme concentration must not be high, since the more enzyme concentration, the more glycerol concentration a sample could have, bringing out star activity. Besides, buffer temperature and time of reaction are important parameters that will enable researchers to know how to add reagents, what is the appropriate concentration of reactants, and what restriction enzyme will be added at first.